welcome our fourth candidate, Antoni Gralak.
Hello.
Welcome. Here is your thesis. Characterization of human transcription factors in a methylation-sensitive context and deciphering their regulatory role at a single-cell level.
Way too long.
I think it took me 180 seconds to say that. You're lucky you don't have to say it in your presentation.
I know, yeah. Right.
It was done under the supervision of Professor Bart de Planck. Are you ready, Antoni?
Yes.
Enjoy.
Thank you. Most of you have probably already heard about cells and DNA. It may not surprise you to hear that your bodies are made up of many, many cells, and that almost all of them contain your DNA. However, every cell in your body has basically the exact same DNA. So how can a cell at your fingertip be so much different from, let's say, a brain cell? To explain this, I would like to give you an analogy. Imagine cells as cities, thriving in complex ecosystems with road networks, power plants, and various buildings. Inside every city, there is a library filled with millions of books and texts that contain detailed instructions on how to build and run the city. A biologist calls this library the cell nucleus, and the books are the DNA. Now, in my analogy, every city has the same library filled with the exact same books. What's different between the cities are the librarians. You see, each librarian has their own reading preferences. Each one decides which books they pick and pass on to the city's architects and engineers. It's ultimately the librarian's choices that shape how the city is built and how it functions. To make things even more complicated, and we are still in the analogy here, librarians work together. So to truly understand a city, we must know how they influence one another. Now, back in real life, these librarians are a specific set of proteins that interact with DNA. And even today, we still don't know what many of them like to read. So how do we find out? Unlike a library, all of this happens, of course, on an incredibly small scale. In my PhD, I therefore use something like tiny pinches with which I can capture the exact moment when a protein reaches for DNA. Then, with a set of experimental procedures, I can identify the DNA that was grabbed. And that way, I learn what the protein likes to read. And I obtain valuable information about where on the DNA it usually likes to sit, and also with which other proteins it typically interacts. With this knowledge, we can better understand how cells work and also why they stop working in certain diseases. Or, in other words, and to go back to my analogy, if a city is dysfunctional, which librarians would you fire and which would you hire to repair the damages? Thank you very much.
Thank you.